Today we're going to go over the answers to the Chapter 12 and 13 study guide. So the first question wants us to use the diagram with the mice to answer the questions. So number 1 says write the letter or letters that would represent a mouse dying. And so remember, you need to look at the bacterial strain that the mouse was injected with. So if the mouse was injected with the virulent strain or with the rough strain and the heat-killed smooth strain together, either of those cases would result in the mouse dying. So the answer to number 1 is B and D. Number 2 says write the letter that would represent the mouse living. So remember, when the mouse lived, they were injected with the non-virulent strain, which means that it doesn't cause disease, or they were injected with the heat-killed smooth strain, and because the bacteria, when it's heat-killed, is no longer alive, it can't cause illness by itself. So the mice lived at letters A and C. Number 3 says, what is this? So we're referring to this whole picture. So this entire thing is a bacteriophage or a virus. Number 4 says, what does it infect? So if you remember, a bacteriophage is a virus that infects bacteria. Number 5 says which part of it contains the genetic material. So if you look at all the different structures of the bacteriophage, you'll notice that inside of the head of the bacteriophage, you will find the DNA, which is the genetic material. So the genetic material is found in the head of the bacteriophage. And then number 6 says who used it in their experiment. So if you remember, there was 2 scientists and they used radioactive isotopes to label the parts of a bacteriophage. So they labeled the DNA with radioactive phosphorus and they labeled the proteins with radioactive sulfur, and the scientists' names were Hershey and Chase. Number 7 says what molecule did Avery conclude causes transformation? So remember, Avery did an experiment where he used enzymes that broke down either proteins or DNA, and what he discovered is that if you broke down DNA, that transformation could no longer occur. So he discovered that DNA causes transformation. So the answer is A. And then the next few questions, we're going to use this diagram to answer the questions. So number 8 says, what molecule is this? And you should recognize that molecule, that double helix shape, as a DNA molecule. Number 9 says, where is it located in eukaryotes? So remember, eukaryotes are cells that, like humans, have animal cells, plant cells. And in eukaryotes, it's important to remember that DNA is located in the nucleus. Number 10 says what pairs with guanine in the above molecule. So if you remember, guanine is represented by the letter G, and it pairs with cytosine, which is represented by the letter C. Number 11 says the percentage or amount of adenine always equals the percentage of, and remember that's thymine, or T, in the above molecule, which is, again, DNA. Then we're going to use a new diagram to answer questions 12 through 15. So number 12 says what process is shown in the above diagram. And you'll notice that there is 2 strands of DNA that are being separated from each other, and those are the template strands of a DNA molecule. And you'll notice that there's 2 new strands being created. So the name of this process is DNA replication. Number 13 says, what is the name of the enzyme that unwinds the DNA? So when DNA unwinds, it gets unwound in the direction of the red arrow that I just drew, and that process is carried out by an enzyme called helicase. And helicase unwinds the DNA molecule so that the 2 template strands are open for replication. And then number 14 says, what is the name of the enzyme that adds base pairs to the strand? So when you're building that new strand, that copy, the enzyme that adds each nucleotide to those new strands is called DNA polymerase. And lastly, number 15 says what nucleotide will be added next to the top strand. So it's going to be across from the letter T, and anytime you have a letter across from T, it's going to be a letter A in a DNA molecule. Next, we're going to move on to talk a little bit more about RNA and protein synthesis. So we're going to do some matching. So there's three types of RNA. There's mRNA, tRNA, and rRNA. Number 16 says transfers amino acids to ribosomes. And so the RNA molecule responsible for that is letter B, that's the tRNA or transfer RNA. Number 17 says brings the information in the genetic code from the nucleus to the ribosomes. That job is carried out by A, the mRNA, because M stands for messenger RNA. And number 18, this forms part of ribosomes. That's C, the rRNA, because that lowercase r stands for ribosomal RNA. Next, we're going to compare DNA and RNA based on their structural differences. So first, we're going to look at the number of strands. So DNA is a double-stranded molecule, so it has 2 strands. RNA is a single-stranded molecule, so it has 1 strand. Then, they do have slightly different bases, right? So DNA has adenine, guanine, cytosine, and thymine. And that's unique to DNA because RNA has adenine, guanine, cytosine, and uracil. So the uracil and the thymine are different between the DNA strand and the RNA strand. And then last, they have different sugars. DNA has a deoxyribose sugar, while RNA has a ribose sugar. Next, we're going to move on to the matching section. So transcription is letter B, that's making RNA from DNA. Translation is letter D, that's using the mRNA code to make proteins. Number 27 is transformation, and transformation is C, and it's when DNA is absorbed from bacteria, from one bacteria to another, and it allows them to express new traits. Number 28, replication, is A, and that's making a copy of DNA from existing DNA. Number 29 is F, and that's an anticodon. It's a series of 3 bases on a tRNA molecule that match the 3 bases that are found on an mRNA molecule. And number 30 is codon, and that's letter E. A codon is a series of 3 bases, or 3 letters, on an mRNA molecule that code for a particular amino acid. And remember that sequence of codons is what tells us what order amino acids go in to build a new protein. Next, we'll do some kind of fill-in-the-blank questions. So we're going to use this DNA strand, which is called the template strand, to answer questions 31 through 34. So our template strand is this sequence, CCT AGA. So first, number 31 is asking us to write the complementary strand made from replication. So if you have a template strand and you're making a copy of it in DNA replication, the new strand that you're making, or that complementary strand, is going to have the sequence G, G, A, T, C, T. And that's because, remember, C always matches with G, and T always matches with A, and then if we have AGA, that will give us TCT. Number 32 says write the mRNA strand transcribed from the original strand, from that template strand. And so we're going to use mRNA codons, or mRNA nucleotides, instead of DNA nucleotides. So in RNA, we still have G, so we'll have G, G, A, but then we're going to have U, C, U, because the U replaces the thymine in an mRNA strand. Number 33 says what 2 amino acids does this code for? And so remember, you're going to use that amino acid chart that's on the last page of this document, and you should have discovered that GGA is the code for a glycine, and you can abbreviate that GLY, and you can use UCU to find serine, and that's also an abbreviation. You can write that down. Number 34 says what are the 2 tRNA anticodons that match the mRNA codons? So if the mRNA sequence is here, the anticodon is going to be, again, those complementary letters. So it should be C, C, and then U, because remember we're using the letter U because we're making mRNA, and then it should be A, G, A. So that's what you'd find on the tRNA, and those are the anticodons that match with the codon from the mRNA sequence. The last few questions here are about some of the different types of mutations that can occur in a DNA sequence. So before the mutations occur, this is the original strand that is unmutated. And then you can see in A, B, and C that there are some different things that happened. So number 35 is when you have a frame shift mutation. And a frame shift mutation means that the codons are read differently because a letter has been added or subtracted. So the two different frame shift mutations you can see are represented by letters B and C, and that's because this one is missing a letter and this one has added a letter, and that changes the sequence of the codons. Number 36 says which letter or letters point to an insertion. And so an insertion is only when a letter is added. And so you can see that in letter C, we've added a letter. And so that is the insertion. Number 37 says which letter or letters point to a substitution. So a substitution occurs when you trade one letter for another. So you can see that has occurred here. And so then we know that letter A is a substitution. And then number 38 says which letter or letters point to a deletion. And so a deletion is represented by letter B, where this letter G has been completely removed from the sequence. And finally, we can review how to use a codon chart. So let's say, for example, that you are asked to find the codon, or the amino acid that goes for the codon ACG. So then we're going to use our letters. So if the first letter is A, we know it's going to be in that row. And if the second letter is C, we know it's going to be in this box. And then if the third letter is G, we know it's going to be in this row. And so our final answer is threonine, right? So that's how you use that chart. The other chart is similar. So if we try to find ACG over here, we're going to start with the letter A, and then we're going to go with the letter C, and then G, and then that gives us threonine. So that's how you use the different codon charts. They give you the same answer, but in a slightly different way.